So do you have any other question in chapter 9's homework or chapter 9's the class? Do you have any other question? Okay, so let me start chapter 9. So chapter 9 is the bacterial and viral genetic system. Okay, so chapter 9 is composed of the 4 subjects starting from the bacteria and virus have an important role, and also we will learn the several important concepts is like this. Okay, so this is the first subject. Okay, so so all prokaryotes are unicellular and lack membrane-bound nucleus, and the membrane-bound organelle is like this. Okay. Okay. Another important thing is that this is the characteristics of bacteria. The prokaryote. Okay. So so this one is important. Reproduce asexually. So because of they reproduce asexually, so maintain their characteristics from generation to generation. So it means that they are genetically pure strain. And also because they are haploid, their status is haploid. So recessive mutation are not masked. Okay. For example, in our eukaryote, we are diploid. So in this case, recessive are the phenotype of the recessive mutation can be masked, but in the bacteria, they cannot be masked because they only have the one. Okay, and this is characteristics. Okay, and also there are 2 kinds of bacteria. First one is prototropic. The next one is the auxotropic. So prototropic means that they can capable of synthesizing all of the metabolite from the inorganic material. However, auxotrope, so they have lost the ability to synthesize some organic compound. Okay, so they need a nutrient. So this is we call it the auxotrophs. And we also covered what is the minimum medium and what is complete medium. Okay. And also bacteria genome, especially bacteria, they have their own chromosome. Their own chromosome is located like this. And they also have the circular DNA. We call it as plasmid. Okay, especially the episome, we call it as F plasmid. They look like this. So they contain gene that regulate lots of the function is like this. Okay. Now, from now on, this is very important. Gene transfer in bacteria. So they are composed of the 3 methods. First one is conjugation. Second one is transformation. Third one is transduction. Conjugation means that direct transfer of DNA from one bacteria to another. Transformation means that bacteria takes up the pre-DNA. Transduction means that virus take the DNA from one bacterium to another. A little bit different. Okay. So let me start from this experiment. So some of them, they don't have some gene. Some of them, they don't have some gene, but they mix to each other. So finally, they found that one bacteria has all of the needed gene is like this. Okay, it means that the conclusion is that genetic exchange and recombination happens between the two mutant strains. Okay, we call it as F plus cell, means that donor cell containing the F vector, and the F minus cell is recipient cell lacking the F vector. Between two cells, they have the sex pillars. This is connection 2. Okay. And based on Davis YouTube experiment, we find that they need direct contact between the bacterial cell. Okay, because this membrane, through this membrane, media can be going here and going here, but bacteria cannot penetrate this filter. So in this case, there is no transforming, there is no recombination. So it means that genetic exchanges requires direct contact between bacterial cells. Okay, and also this is also important. So F plus cell is donor and F minus cell is recipient. So they have the F vector. So gene transfer is starting from the F vector. Okay, through they have connection tube is like this. At that F plus cell, still F plus, and recipient cell become the F plus cell is like this. Okay, and also additional one, we have to know HFR cell. HFR cell, we call it as the high frequency strain. So it means that the F vector is integrated into the genome. So this cell, we call it as HFR cells. Okay, and also conjugation happens between the HFR cell and the F minus cell. Okay, and gene transfer starting from here. Okay, go there. At that, usually the genetic recombination happens. Okay, so finally it will make the HFR cell. Still, this one is F minus cell. But this F minus cell, some of them is recombined to each other like this. Okay. Final one we have to know in conjugation, F prime cell. F prime cell means that the F plasmid carrying some bacterial gene is like this. Okay, for example, this F plasmid has some bacterial gene, especially in this case, RAC gene is like this. So this gene comes from the bacterial chromosome. So this cell, we call it as F prime cell. Okay, so between the F prime cell and F minus cell, the conjugation also happens. Okay, starting from here. Okay, so F prime cell still F prime cell, and the F minus cell becomes the F prime cell. Okay, so in this case, so usually the bacterial cell is the haploid. They only have the one chromosome. However, in this case, the F plasmid carrying the bacterial gene, some of bacterial gene. So in this case, they are partially diploid. So we call it as merozoite, merozygote. Okay, they are partially diploid. Okay. So they summarize here like this, and also they summarize it like this. Okay, in our homework, okay, okay, let me explain more. Okay, okay. So we can map the bacterial gene with interrupted conjugation. Okay, this is important. So distance between gene is measured by time required for DNA transfer from HFR cells to F- cell. Okay, so after conjugation is happen, so we interrupted. So conjugation was interrupted at regular intervals. So sometimes we use sonicator. Okay, since sonicator, it has a higher the frequency of the energy, so the conjugation can be interrupted. Okay, okay, so based on this one, so starting the 8 minutes later, so sonication is happens. Okay, 10 minutes later, also sonication is happens. So 8 minutes later, so the another azid azid gene can be shown up. So this is azithromycin resistance gene. So this bacteria has the azithromycin resistance. Okay, so initially the F- cell, they don't have the azithromycin, but 8 minutes later, the azithromycin show up. And also 10 minutes later, another tone bacteriophage T1 resistance gene is shown up. And also around 16 minutes later, another LAC+ gene gene is also shown up. Finally, the 25 minutes later, the GAR+ gene is shown up. Okay, so based on this one, because the transfer time indicate the order and the relative distance between the genes. Okay, starting from the the gene order is AZI and tone, LAC, and GAR. Okay, so between the RG and tone, there is a 2-minute difference. 2 tone and LAC, there is 6-minute difference, 6 difference, and also LAC and GAR, there is the 9 minutes difference. So this will be the genetic distance is like this. Okay, so in our homework we cover this one. Okay, this experiment is exactly same as the previous one. So they use the HFR strain, which has all of them positive. Okay, and F prime cell, F minus cell, they are all of them, this gene is negative. Okay, based on this one, MAL+ gene is shown up around 22 minutes. And at that XYL gene is shown up, and the MTL gene is shown up, and methionine gene is shown up is like this. So based on this graph, this one, we can make the gene order starting from the MAL, XYL, MTL, and methionine. So the distance between this one will be the 7 minutes, 1 minute, and 9 minutes is like this. Important. Okay, you have to know this concept, okay, to solve the final term exam. Okay, so conjugation, you have to know this concept, okay? Very important. Okay, this is genetic class. This concept is very important to the genetic class, okay? Okay, And also this is another important concept, directional transfer and mapping. So usually the HFR strain has F factor is integrated into the chromosome. Okay, so F factor is integrated at different sites and also different orientation. But however, gene transfer begins within the F. So, and also, so for example, in this case, gene transfer is starting from here, after leucine, threonine, thymine, histidine, GAR, like this. So in this case, gene transfer is starting from here, after THL, threonine, leucine, like this. Okay, so we can determine the direction and starting point of the gene transfer is like this. Okay, so based on this one, so this one is one HFR gene. Okay, so this is the appearance of gene in F- cell. So B plus is shown up 3 minutes later, and 5, okay, and 16, 27, 59. In the HFR, when we used HFR2 and F-, the conjugation is happens after E plus is starting from the 6 minutes, 24, 35, like this. So when we conjugate the HFR3 and the F-, D plus is starting 4, 15 is like this. Okay, based on this information, you have to draw the structure is like this. Okay. Okay, is important. Okay, this concept is important. Directional transfer and mapping. Okay, usually the gene transfer begins within the F vector. So they have orientation like this. Okay, it's very important. Okay, so you have to know how to construct a genetic mapping based on this data. Okay. Okay. Now we will go to the transformation. So transformation means a bacteria takes up the DNA from medium. So this one is the DNA. So bacteria is takes up the DNA. At that transformation, recombination happens. So this one will be the transformed bacteria is like this. Okay, and also this is also very important. So recipient cell and transformant and the co-transformant. Okay, co-transformant means the cells that are transformed by 2 or more genes. And also this concept is very important. So transformation can be used to map the bacterial gene. Okay, so the rate of co-transformation is inversely proportional to the distance between them. Okay, for example, A and B, they are very close to each other. It means that the rate of co-transformation of this one is higher. Okay, for example, in this case, for example, in this case, the recipient is A minus, B minus, C minus, like this. Okay, whenever they take up A plus, they will become A plus, B minus, C minus. When they take up B plus, they will become A minus, B plus, C minus, like this. Okay, so when the co-transformation happens, it means that their distance is very close to each other. Very important. We have to know this concept. So in our homework, we also have the 25 So the genotype is A plus, B plus, C plus, D plus, E plus, is used to transform a strain with genotype A minus, B minus, C minus, D minus, E minus. So pairs of genes are checked for co-transformation, and following results are obtained. Okay, in this case, co-transformation happens. Co-transformation A plus B plus does not happen. It means that the distance between A plus B is very far away from each other. For example, A and D, their co-transformation happens. It means that they are very close to each other. Okay, on the basis of this concept, you have to write the order is like this. But important thing is that if you solve the question very carefully, so B and C, so B can be here or B can be here. So the orientation of B plus C plus is relative, cannot be determined based on the data is provided. So we have to write down here like this, okay? So D, A, E, C, and C and B, we cannot determine the location. So in this case, we can write down like this, okay? Very important. Okay, you have to know this one. The rate of co-transformation is inversely proportional to distance between genes. Okay, this one is very important. Okay. Okay. Let me go to the transduction. So transduction means that virus is involved in gene transfer. So bacteriophagy is a virus that can impact the bacteria. They have the lytic cycle, and they have the lysogenic cycle. So virulent phagy means that reproduce through lytic cycle and always kill the host cell. So finally, they kill the host. This is bacteria, and this virus finally kill the bacteria. Okay, so it means that the clear region. So usually in our plate, so we add bacteria. So their color is just their color is not their color is a very gray. Is not clear, but in this clear region, we call it as a plaque. So this is a clear patch of lysed cells. Okay, so on it means that in this area, bacteria is died. So based on this one, here there are lots of bacteria here, but this region is bacteria is died. This the circle region, this region is bacteria is died because the bacteriophagy finally lysed the bacteria. Okay. Okay, so it means that this experiment, so they have filter, but the recombination happens. It means that virus can penetrate this filter. They can go here and there, here and there. It means that bacteriophagy is the agent of the transfer. Okay, genetic exchange did not take place through conjugation. So based on this experiment, we can say that bacteriophagy is the agent of the transfer. Okay, so this is the generalized transduction. Okay, so for example, this party is finally they lysed the bacteria. Okay, so it means that this bacterial party has the bacteria. So this is bacterial chromosome. So this bacterial party has bacterial gene. After it can transfer the bacterial gene to the next recipient cells. After that, recombination happens. Okay, so it becomes the transductant cell. Okay. Okay. And also, this one is very important. This concept is very important. The rate of co-transduction is inversely proportional to the distance between genes. Okay, so in this case, co-transduction happens. It means that the gene distance between the A and B, they are very close to each other. Very important. So, and also this is also another the recombination frequency. Okay. So an E. coli is infected with two different strains. One has the H plus, R minus. The other one has the H minus and R plus. And usually the between them after impact the bacterial cells. Okay, usually the recombination happens. So they will make this kind of combination. Okay, so usually this one, the flock number, the largest one will be the parent. No recombination. This case is no recombination. So H minus and R plus means that clear and small. And H plus is cloudy, and R minus is rosy. R plus is small, and H minus is clear. Okay. So in this case, they are very clear, but their size is small. So in this case, their genotype will be H minus R plus. Okay. Okay, so in this case, it's very clear. It means the H minus and very large. That's why they are R minus is like this. So in this case, they are not clear. So this will be the H plus cloudy. And this is very large. And their genotype is R minus is like this. And this one is also is not clear. So it will be H plus. But it's very small, it will be R plus is like this. It's okay, based on this one. So this one will smallest number will be the recombinant progeny. So based on this one, you have to calculate the recombination frequency. And also we have this kind of question, number 13. Okay, one party has the A minus, B minus mixed with wild type party, A plus, B plus. So we have this kind of the number. So this one will be the non-recombinant, largest one, and this one will be the recombinant. So based on this one, we can calculate it is like this. So in final term exam, we need calculator, okay? And also, we also did the one party has HSTC, The other fuzzy has the wild type H plus, STC plus. So based on this one, so we have to know what is parental, non-recombinant, recombinant, and double recombinant. Okay, based on this one, we can first we have to decide the order. So ST is center, and also determine the map distance between the gene, and we also can calculate the coefficient or coincidence, and also we can calculate the interference is like this. Okay. Okay. Do you have any other question in chapter 9? So chapter 9 is okay. We cover very important concept. Okay. So we have to understand the conjugation, and transformation. This is the transduction. This is also transduction. Do you have any other question? Ronika, do you have any other question?
No, I'm good, thank you.
Okay, so you have to understand this concept, okay? So already in chapter 7, we already covered the how to order the direction, okay? You have to know this concept, okay? Very important. This is very important. This everything is important. Homework is very important to us, okay? Okay, do you have any other question? Okay, so we will go to the chapter 10. So in chapter 10, do you have any other question in chapter 10 course or the homework? 김건영, 수업도 안 들어오고 이 학생. 김건영, 이 학생 어떻게 된 거지? 김성환, 아까 있던데.
네.
김건영 너
네.
친구 아니야?
잘 모르겠습니다.
이것도 한번 연락해봐, 알겠지? 수업도 안
네.
들어오고 이렇게 하는데. 수학과 주채연 들어왔어? 장준원 들어왔어? 아까 안 들어왔던데? 최민준 안 들어왔던데 아까? Okay. Okay, so we will cover chapter 10. Do you have any other question? Okay, so we will start chapter 10. Chapter 10 is DNA, the chemical nature of gene. Okay, so first one we will cover the genetic material possesses several key characteristics. And we will also learn the DNA and RNA structure. Okay. Finally, we learned some DNA special structure in DNA and RNA. So this one is the genetic material possesses several key characters. Okay, they have complex information, and also they replicate faithfully, and they also encode phenotype, and they have the capacity to vary. So in our homework, we cover this one. First one, what are the general characteristics most the genetic material possesses? They have the 4 important thing is like this. Okay. And also this is the DNA structure. The important thing is that Chagas floor. The Chagas floor means that the regularity in base ratio. means that the base ratio A and T is exactly same, and G and C is exactly same. Okay, and this is the Chagap's rule, the regularity in base ratio of DNA. The amount of adenine is equal to the amount of thymine. A and T is same, and the amount of guanine is equal to the amount of cytosine. Okay, so A to T is nearly 1, and G to C is nearly 1. So it means that A plus G divided by T plus T will be also is like this. Okay, we have to know to understand this one. So this is also mentioned of Chagap rule. Okay, so you already compute the ratio between them, and also you can already determine the ratio. So this one will be nearly 1. This one is the different, is like this. The important thing is that A and T is nearly same, and G and C is nearly similar to each other. Okay, and also this number 32, if the thymine is 15, what are the percentage of other base pairs? So thymine is 15 means that adenine is 15. So based on this one, the guanine and cytosine will be 35, is like this. And this one is very, very important. Okay, so virus 1, they have U instead of T. It means that they are RNA virus. And also A and U, they are same, and G and C is they are same. It means that they are double-stranded virus. How about this one? They don't have the this one is a double-stranded RNA virus. So this one is a double-stranded RNA virus. They have the U. In this case, they don't have U, but the A to T is exactly the same 23. C to G is exactly 16. It means that they are double-stranded DNA virus. How about this one? 3, they don't have U. It means that they are DNA virus. However, A to T is different. C to G is different. It means that they are single-stranded RNA virus. Single-stranded DNA virus. So it means that they are single-stranded DNA virus. Number 4 is they have U. It means that they are RNA virus. The ratio between A and U different. The ratio between C and G are different. It means that they are single-stranded RNA virus. Okay, Chagap rule is very important. Okay. And also this one is Grips experiment. Okay, it means that based on this one, substance in the heat-killed bacteria transform bacteria into live and virulent bacteria is like this. Okay, you have to know. Another one is that Avery and McLeod and McCarthy's experiment. So one of them they added RNAs, one of them they added proteins, one of them they added DNAs. So in this case, there is no recombination happens. There is no cell. But in this case, type S and the type R, they are coexisted each other. Okay, so it means that the DNAs destroyed the genetic material. So it means that because the only DNAs destroy the transforming substances, so it means that transforming substance is DNA. Okay, and also we have this kind of question in our homework. So this one. So one is okay. So we will see it later. Okay. Okay. And also we use sometimes we use the virus. Okay. So they did the transduction. So finally, the bacterial cell lies and releasing the progeny process is like this. Okay, so this is Hershey and Chase experiment. Okay, whenever we use P32, it means that it can be incorporated into the DNA because DNA has phosphate. Whenever we use the and also protein has the methionine. So protein, amino acid, methionine, and cysteine, they have sulfur. Okay, so whenever we impact with the isotype with F35, it means that this one can incorporate into the amino acid. Okay. Okay, so you have to understand this one to solve our homework number 24. So based on this one, tube 1, the presence of radioactivity in the cells. But this one is protein coat. It means that they are labeled with S35. This one is also S35. However, tube number 1, 4, 5 is labeled with P32. Okay. Okay. And also, this is DNA structure is the double helix. So Watson and Crick find DNA is their structure is double-stranded and helical structure. Okay. Okay, so and also their structure is anti-federal, starting from the 5' to 3, 5' to 3 is like this. Okay, so their structure is anti-federal DNA structure. Okay, 5' to 3', starting from 5' to 3' is like this. Okay. And also this one, our secondary structure of DNA. So you have to know what is bDNA and what is aDNA. So bDNA, it happens nearly 90% of DNA is existed as bDNA, and maybe less than 10% is existed is in aDNA, and zDNA is not happens very well, less than 1%. Okay, so you have to know what is bDNA and what is the aDNA to solve the final term exam. Okay, so bDNA is right-handed. So, and also 0.34 nanometer between the adjacent base pairs and 10 base pairs per turn. It means that 3.4 nanometer per turn. Okay, you have to memorize it, okay? And the aDNA is also right-handed. This is bDNA is the perpendicular to the axis. but aDNA is not perpendicular. And also, this is the value you have to know, 0.26 nanometer, and they have the 11 base pairs per turn. So it means that they are 2.8 nanometer per turn. You have to memorize it, okay? So we have this, there are several questions so you have to know, to solve this one, you have to know importance 0.34 and 10 or 3.4. You have to memorize this one, okay? Okay. And also, this one is exactly the same as you have to know about the bDNA. So bDNA has 1 million nucleotide pairs, means that how many turns? Okay, so it means that 1 million divided by, so 1 turn is composed of bDNA, 1 turn is composed of 10 nucleotide pair divided by 10. Okay, how about the aDNA? aDNA is per turn, they have 11. Okay, so 1 million divided by 11 is okay. Okay, and also you have to know, so this is the pathways starting from the DNA is go to the RNA, transcribed, after that RNA can be translated. Okay. Okay, and also RNA, their structure is they are single-stranded. Sometimes, so in this case, so A and T, they compare to each other, they bind to each other, so they made the inverted, okay, hairpin structure is like this. So in this case, A and T, G and C, they made a stem, and another structure they have loop is like this. Okay, so this is the RNA is composed, they are single-stranded, but sometimes they have hairpin. Okay, their structure is like this. And also we covered HDNA, and also we covered DNA maturation. Okay, so in this today's lecture, DNA maturation is methyl group is added into the nucleotides. So this is DNA maturation is related to the gene expression. So this affects the three-dimensional structure. If they have lots of maturation, so chromosome structure is very packed. So it means that transcription does not happen. However, if the methylation is not there, so the chromosome structure is very relaxed. So it means that transcription happens very well. Okay, so there are lots of mechanisms in the DNA methylation, but today I will not cover. And next semester I open the epigenetics. At that time I will explain some details information. Okay, today's lecture, just the DNA methylation is related to gene expression. Okay, whenever they have methylation, transcription does not happen very well. It's like this. Okay, and our homework. So we made some kind of helping structure is like this. Okay, UA and AU like this. Okay. Okay. Do you have any other question? Chapter 10. Do you have any other question? 양예은.
네.
Yes.
네.
지난번에 자고 있던데? 양예은 지난번에 수업 시간 자고 있던데?
네, 맞아요.
Okay, so 신은나, 너 수업 잘 듣고 있어? 오늘 회사 출근하는 날 아니잖아. 신은나, 수업에만 들어오고 수업도 안 듣고 있는 거야? 쉬는 날? 아, 출근했구나. 오케이. 오케이. 다들 박승아 수업 잘 듣고 있어?
네.
왜 이거 카메라 안 켜? 오케이. 김민주, 두 김민주 잘 듣고 있어? 나노바이오.
네.
오케이, 숙제 잘 내고 있고 숙제 잘 냈지 맞지? 오케이. So, okay, before going to chapter 11, okay, so maybe we have some break. Okay. I think now most of the students is the third grade. Most of the students is third grade. Okay. So it means that you have to decide what you want to do. Okay. You have to do it. Okay. Okay. And also I found that there is one way, another way. All of students want to go to the apply to the job, but also you can go to the graduate school, or you can go to the graduate school in the America or any other place, other countries. Okay. Ronika, I will speak in Korean briefly, okay? So this is not related to the course, okay? 그러니까 뭐 여러 가지 길이 있고요. 여러분들 지금쯤 이제 선택을 하고 그에 대해서 매진하면 될 것 같아요. 알겠죠? 이지수, 알겠어? 그러니까 단 선택을 하고 나면 절대로 후회해서는 안 돼요. 저는 어떻게 하냐면요. 선택을 하고 나면 그때 내가 내린 선택이 그 당시에 내가 할 수 있는 최선의 선택이었다 생각하고 절대 후회하지 않거든요. 그러니까 후회하지 말고요. 알겠죠? 그다음에 뭐 유학 가는 것도 생각해 보세요. 유학 가는 것도 좋은 기회니까요. 인천대 학생들이 유학을 거의 안 가려고 하더라고요. 그러니까 유학 가면 좋은 대학교 많이 갈 수 있습니다. 최문석.
네, 교수님.
그러니까 미국에도 바이올로지 디파트먼트가 있을 거잖아, 맞지? 미국 애들도.
네, 맞습니다.
걔들은 1등부터 30명 있다고 치면 1등부터 10등까지 말할 것 같아.
잘 모르겠습니다.
걔들 1등부터 10등까지는 무조건 메디컬 스쿨 아니면 덴탈 스쿨 아니면 파머시, 수의대 그런 데 다 가요. 미국에는 싹 다 그게 이제 학부에 있는 게 아니고 석사에 있으니까 이제 다 글로 가요. 그러면 11등부터 20등까지는 뭐 하죠? 최무석 말할 것 같아. 대부분 걔들은 취업을 해요. 왜냐하면 미국은 학부가 엄청 비싸거든요. 그래서 걔들은 빚이 엄청 많아요. 취업을 다 해요. 그러니까 미국 대학은 누가 갈까요? 미국 대학을 아무도 가지 않아요. 거의 가지 않아요. 그러니까 상대적으로 우리 외국인들이, 저도 미국 대학은 존스 홉킨스 의대 나왔는데 가면 대학은 싹 다 외국인들이에요. 다 인도 아니면 중국 아니면 한국, 다 한국 애들이에요. Very easy to go to the Ivy League, Ph.D, Ivy League School. 그다음에 미국은 학비가 들지 않아요. 생활비를 다 받고 다녀요. 제가 나온 다 나와요. 존스 홉킨스, 스크로브 메디슨, 스타이펀드 이거 치면 있잖아요. 다 나와요. 그러니까 미국은 그러니까 애니얼 튜이션이 58,000불인데요. 다 이건 다 공짜예요. 대학원에 가면 다 공짜예요. 그다음에 34,910불 해가지고 34,910불을 1년에 월급으로 받아요. 모든 대학원생들이. 제가 갔을 때는 24,000불이었거든요. 그때 제가 20년 전에 갔을 때는. 그러니까 살만한 거죠. 오케이. 그러니까 투이션은 공짜고요. 메디컬 인슈런스 해주고요. 그러니까 뭐 여러 가지 기회가 있으니까 기회는 널려 있으니까요. 여러분들이 이제 하기 나름이에요. 알겠죠? 그럼 이제 정지윤.
네,
미국 대학원 가려면 어떻게 해야 되겠어?
영어를 잘 해야 될 것 같습니다.
그 전에 제일 해야 될 게 있습니다. 미국 대학원을 가는 가장 좋은 방법은 나한테 상담을 하는 거예요. 입시 컨설턴트한테 상담을 받으면 금방 갈 수 있습니다. Okay? So if you are interested in study abroad, please let me know. Okay? Okay, so I will teach you how to prepare the Ph.D. school in the America. Okay? So Ronika, if you are also interested in the study in the America, please let me know. I will teach you how to prepare the Ph.D. school in America. Okay?
Thank you.
Okay. Okay, so now we will go to the chapter 11. So do you have any other question in chapter 11? Okay. Chapter 11 is chromosome structure and the organelle DNA. Chapter 11 is composed of 4 subjects, starting from the DNA and also recover eukaryotic chromosome, and also they have sequence variation. Finally, we will learn the mitochondrial DNA. Okay, so they have a supercalling. Usually the one ton, they are composed of usually the DNA is the bDNA. So it means that one ton, they are composed of the 10 nucleotide pair. So if they have more nucleotide pair, they become the supercoiling. So if DNA is unrotated, if they are less than 10 base pair, they will become the negative supercoiling, like this. So based on this one, they have to like this. Okay, so you have to calculate it, and it means after that you have to decide this is positively and negatively and relaxed like this. Okay, and also this is bacterial chromosome. They are composed of the single circular DNA molecules. Okay, they don't have the nucleus. Okay, and this is the eukaryotic chromosome. Their basic starting point comes from the nucleosome. So nucleosome is a fundamental repeating unit of the chromatins. So they have the 8 proteins. 2 set means that they have 2 H2A, 2 H2B, 2 H3, and 2 H4. So usually the DNA, they are 104 or 107 base pairs. Okay, so it means that this DNA laps the histone proteins 1.651 times and 65 times. Okay, and also, so our homework, we cover this one. So what is composition of nucleosome? So this is their structure is like this. And also chromatosome, so nucleosome with histone H1. So this is a chromatosome. Okay, so it means that one chromosome, they have 8 histone protein plus 1 H1, total 9. Okay, you have to understand this one. So finally, we curve in our homework, we asked about this concept. How many histone proteins there are in this DNA? So in this case, we have to multiply 9 histone, okay? The nucleosome number multiplying 9 histone, okay? And also, chromata, they have the additional 20 base pairs DNA and the link DNA. So they have this link DNA. Their size is like this, okay? So in our homework, we also cover this one. I cannot find it, okay? Okay, so, okay. And also this is our chromosome structure, starting from the 30 nanometer fiber, and they will become the chromatid is like this. And also this is the euchromatin structure and the heterochromatin structure. Okay, so in our homework, we compared each other. Euchromatin is less condensed, heterochromatin is more condensed, is like this. So usually euchromatin is located in chromosome arm. Okay, heterochromatin is there located in the centromere and the telomere region. Okay. Okay. And also we cover the polytene chromosome, especially in the Drosophila, and especially in the salivary gland of Drosophila. All of chromosomes combined to each other. So they made the polytene chromosome. Okay, so if you see here, they appear striped with alternate dark and white band. Dark and white band. So usually the light band, this region, chromatin is relaxed, and this the dark region, this region chromatin is complexed. Okay, and also chromosomal pulp. Sometimes they have chromosomal pulp. So in this case, chromosome is very relaxed. It means that transcription is taking place very actively. Okay, and also this is so we cover this one. What is polytene chromosome and what is chromosomal pulp? So and we also covered what is difference between the euchromatin and heterochromatin is like this. Okay. And this is chromatin structure. Okay, so it means that changes in chromatin structure is determined by DNA's one sensitivity. So DNA's one sensitivity is correlated with gene expression. For example, if there are open chromatin structure, after we treat DNase 1, it can degrade DNA. However, in condensed region, still the DNase 1 cannot penetrate inside, so their structure is same, is like this. So based on this one, so in this chicken has the you have to know this one, embryonic globin gene, and they also have the adult globin gene is like this. So post 24 hours, there is no sensitivity region. It means that there is no gene expression of U, there is no gene expression of alpha T and alpha A. So it means that, and also the 5 days, they are very sensitive in the U region. It means that 5 days, the U gene is actively transcribed. Okay, in the 14 days, alpha D and alpha A, they are very DNase 1 sensitive. It means that alpha D and alpha A is actively transcribed. Okay, you have to know. So we already solved this question. Okay, very important. Okay, very important to us. Okay. And also this is the chromatin structure is also changes the color in the coat color in the mouse. Okay, so today I will not cover in details. Okay, so changes in chromatin structure also changes produces different coat color in mice. Okay, but if you are interested in details, I will explain it next semester. We have epigenetics class. Okay, and also this is centromere structure. And this is, we cover the telomere structure. At the end of the chromosome, we have the telomere. And the C value means that the amount of the DNA per cell. Usually the bacteriophagy, they are very small. And this is our Homo sapiens. So our genome size is like this. However, some salamander, another plant, they have the larger one. We don't know why. And most of the gene in the salamander, they are repeated. And also they have lots of pseudogene. So we don't know why, okay? Also C value means that amount of DNA per cell, okay? And we also mentioned that what is C value, okay? Okay, so and we also have to know what is TM. So Genet ratio. So usually this is a DNA is the double-stranded. After we increase the DNA, finally they will become the two single-stranded. Okay, TM means that the midpoint of the melting curve, this is recorded as TM. So their TM will be around the 70 centigrade. If they have the last of the GC content, between the GC, they have three hydrogen bonding. Between the A and T, they have 2 hydrogen bonding. So it means that GC content is higher. It means that TM is also higher. So we cover this one in our homework. So which one is they have a lower melting temperature? Okay, so it means that this one has a higher GC content. This one has a less GC content. So it means that this one. Okay. Also, we have the eukaryotic DNA have gene family and pseudogene. Okay, now is the very important DNA microRNA, mitochondrial DNA, and also chloroplast, they have the DNA. Before we go to the here, in our homework, we cover this one. Assume that the linker So linker is about 30 base pair. 30 to 40. So in this case, we assume that linker is 40. So they have the 6.4 billion base pairs of DNA. Okay, so in the question, I will assume that the DNA, so the nucleosome DNA is composed of around here. So in the question, I will write down the nucleosome DNA, the length will be around 140. Okay, so that one is very easier to calculate. Okay, the chromatome DNA is about 20. The link DNA is 40. Okay, based on this one, you can calculate how many nucleosome is present in the cell, and also you have to calculate how many histone protein. Multiplies 9 is like this. Okay, and also now we will cover very important thing, mitochondrial DNA. Okay, so mitochondria have DNA. So usually our sperm also they transfer their DNA. Also, they transfer their mitochondria. However, sperm's mitochondria is degraded. So it means that almost exclusively from female parents. Important. Also, so duplicative segregation happens. The heteroplasmic cell, they are wild type. They contains two forms, wild type or mutant. Okay, so sometimes they will become the heteroplasmic, sometimes they will become the homoplasmic, and the homoplasmic is like this. So this concept is very important to solve the exam. So this one, okay. I will explain one more. So, okay, so important thing is that mitochondrial DNA. So mitochondrial DNA is maternally inherited. So mitochondrial DNA is identical from generation to generation. Okay. So, for example, the mother has the disease. Second generation, all of the offspring has the disease. In this case, mother also has the disease. In this case, they have 3 children, but they have 3 children have disease, but 1 children is not. They have the disease because of this one. So usually this one also has disease. However, heteroplasmic is happens. So some of them will become the homoplasmic, or some of them they will become heteroplasmic. Some of them have another homoplasmic is like this. It means that their children, some of them will have the disease, but some of them they will not have the disease because of this one. Okay, so based on this one, so this disease is the cytoplasmic inherited trait by the mitochondrial DNA. Okay, and also this one is the, for example, 38. So 38. This is the male, but their offspring will not have this disease because this is the maternally inherited. mitochondrial DNA is maternally inherited. Okay. Okay. So do you have any other question? Okay, so do you have any other question? Okay, see you next Monday. So you need a calculator, okay? Okay, so we go. Okay, so see you next Monday. Okay, bye.
Bye. Thank you. Have a nice day.
You too.
감사합니다.
Okay.
감사합니다.
아.